My name is Scott Biering, and I'm a postdoctoral scholar at the University of California, Berkeley. And I was a finalist for the Michelson Philanthropies and Science Prize for Immunology for my essay on antibodies targeting the non-structural protein 1 of flaviviruses. I got interested in the field of immunology because I came to appreciate that viral pathogens only exist in the face of coevolution with our immune systems. So I wanted to understand how our immune systems recognized viruses as invading pathogens and use that information to produce new therapeutics and vaccine strategies targeting these viral pathogens. My current research investigates how emerging viral pathogens cause severe disease in humans. So we focus on two viral families at the moment, the flaviviruses, which are mosquito transmitted, and they include dengue virus, Zika virus, and West Nile virus, and more recently SARS coronavirus 2. We specifically want to use this knowledge from our research to, one, produce new therapeutic and vaccine strategies against these current viruses, the flaviviruses and the coronaviruses, but we also want to apply general themes to even new emerging viruses that we don't know about yet. My ultimate motivation that keeps me going is really twofold. It's both a passion for alleviating global disease burden with the research that I do, but it's also mentoring a new generation of scientists. The Michelson Philanthropies and Science Prize for Immunology will have a great impact on my ongoing career, both because the funding will help me to set up new systems to understand how viral pathogens cause disease, but also to help me recruit new lab members and get an independent investigation position to continue this interesting avenue of investigation.
Well, hello, everybody. First, I'd like to thank everybody for coming to this event. And I'd like to start by thanking Michelson Philanthropies and Science for allowing us to share our work today. I'm very excited to tell you about some of the work that we've been doing to understand how flaviviruses cause disease and how that knowledge can help us come up with versatile approaches to produce new vaccines and therapeutic targets. And my name is Scott Biering, and I'm a postdoctoral scholar here at UC Berkeley. First, I'm just going to hide these, and then we'll get started. So first, I'd like to tell you a little bit about flaviviruses and dengue virus specifically. These are positive sense RNA viruses of the Flaviviridae family, and they're spread by insect vectors like the Aedes genus mosquitoes. And essentially, the habitat of these mosquitoes mirrors where you see these viruses spread, so often tropical and subtropical locations. And greater than 1/2 of the world's population lives within range of infection of a flavivirus, like I mentioned, they cause a huge disease burden with dengue, Zika, West Nile, Japanese encephalitis, and yellow fever, to name a few, which cause diverse disease outcomes, with dengue and yellow fever believing to be more of a systemic infection, with yellow fever targeting the liver, while Zika, West Nile, and Japanese encephalitis more target the brain. And Zika, of course, causes other complications like microcephaly. Now, very few vaccines are available to target flaviviruses, and no approved therapeutics are available. And one of the complications, especially for dengue virus, to produce a vaccine is the fact that closely related dengue virus serotypes, so there are 4 serotypes for dengue virus, can basically enhance infection of another serotype. And the reason for this is the antibodies that are produced against the envelope protein of dengue virus, they may be neutralizing against a homotypic infection or that specific serotype, but the cross-reactivity may be subneutralizing against a different serotype and may actually end up enhancing infection through allowing further infection of target cells like monocytes by uptake of antibodies complex with virus in an Fc gamma receptor dependent manner, and this increases infection. Needless to say, this has basically made making a vaccine challenging So a major avenue of investigation for us has been to look into alternative targets besides envelope protein. And one such target for flaviviruses is this conserved non-structural protein 1, or NS1. And NS1 is really interesting because it's found intracellularly and is important for replication of the virus. It can be found as a dimer inside the cell, but it's actually also secreted in this hexameric form into the blood of patients from infected cells. And we find that more severe forms of disease, so dengue hemorrhagic fever in this case, are found to have higher levels of NS1 compared to less severe forms like dengue fever in this situation. And this NS1 protein has been shown to act as a direct virulence factor by activating platelets and immune cells, inhibiting complement, or directly triggering endothelial dysfunction and vascular leak, which is a major hallmark of severe dengue infection and even other flaviviruses. So as I mentioned, Flavivirus NS1 causes vascular leak. This has been something we've been showing for some time. And the capacity to cause vascular leak of this NS1 protein happens in a tissue-specific manner. So here I'm showing you an in vivo vascular leak model where you can administer a given Flavivirus NS1 protein intravenously to mice, and then give a tracer dye like dextran conjugated to a fluorescent dye like LX4680, and you can see how much of this dye accumulates in a given organ. So here in red in the brain. And you can appreciate, I hope, that dengue virus, Zika virus, and West Nile virus NS1 causes vascular leak in the brain, which correlates with the disease manifestations of these given viruses, while yellow fever virus causes less leak. If you look in the lungs of mice, you can see most vascular leak for dengue, which is known with severe lung complications. And for yellow fever virus, this causes more vascular leak in the liver, while also dengue does this, while West Nile and Zika does not do this. So the point is that it causes vascular leak, and we've been interested to understand how therapeutics can block this pathology and potentially treat multiple virus infections. So to do this, we took both an immunological and a biochemical approach to understand the structural basis by which NS1 antibodies block this pathology. So we teamed up with Janet Smith's lab at the University of Michigan, who are actually the people to solve the first crystal structure of NS1, which revealed this really unique structure with three distinct domains: beta roll in blue, the wing in yellow, and the beta ladder in red. And one of the first things we wanted to understand is how these unique domains may be conserved amongst flaviviruses and how they may contribute to severe disease. So first, we wanted to have a tool to ask this question. As I mentioned, we wanted to look at how antibodies may block this pathology. So we used a homemade NS1 monoclonal antibody called 2B7, which was raised against DB2 NS1 in our lab some years ago. And what we could show is that this NS1 antibody was actually very protective against a lethal flavivirus challenge. So you can see here that mice that received passive transfer of 2B7 are partially protected from severe disease compared to mice that received an isotype control. This antibody was also protective against vascular leak in vivo and in vitro. So what does this antibody look like in complex with NS1? Well, as I mentioned, Janet Smith's lab, we were able to solve this crystal structure. And you could see here the NS1 dimer in green and the 2B7 fab in blue. And you can see it binding strongly to the beta ladder of NS1, but it binds to the beta ladder in such a way that it comes off in this interesting arc-like projection, which we predict would interfere with this protein from interacting with endothelial cells. So we kind of predict this protective mechanism in two ways. One, that it binds directly to the beta ladder, and the second, that it may sterically hinder other domains like the wing domain. So the resolution of this crystal structure was good enough to see specific amino acids that are interacting with the antibody. So we predicted that some of these amino acids may contribute to pathology of NS1, but then also the wing domain might as well. So we made some NS1 mutants. So A303W, E326, and E327 are all in the beta ladder where 2B7 binds. And then we also made a wing domain mutant, a different domain of NS1. And we could find that these NS1 mutants were all defective for causing endothelial hyperpermeability in vitro in this TIR assay, which measures hyperpermeability across an endothelial cell monolayer. So this predicts that both of these domains are important and suggests this is why 2B7 could be so protective. What was exciting, though, is we found that in red here, the regions of 2B7 beta ladder that were bound to 2B7 were actually fairly conserved amongst many different flaviviruses, leading to the potential that maybe TUB7 could bind to NS1 from multiple flaviviruses. And indeed, when we did these ELISA assays, these direct ELISAs, we could find that TUB7 could bind to NS1 from multiple flavivirus serotypes, as well as many different flaviviruses, including Zika, St. Louis encephalitis, West Nile, Japanese encephalitis of Sutu, Wessel's brawn, Powassan, tick-borne encephalitis, and yellow fever virus. Now, we wanted to see if this high cross-reactivity could correlate with cross-protection and functionality of this antibody. And we could find that indeed in vitro, TUB7, both the full length and the FAB, blocked West Nile and Zika virus NS1 from binding to the surface of brain endothelial cells. And this translated in vivo as well. We could find that TUB7 also offered some protection in a lethal Zika virus challenge of mice. And this provided the first proof of concept that one specific therapeutic could block against severe disease of multiple flaviviruses by targeting a single conserved flavivirus protein. So obviously some of the future directions, which are very exciting, are expanding this further, looking at more different domains in NS1 that are highly protective and conserved, and seeing if you can protect against multiple flavivirus challenge models using one therapeutic. And we're also engineering TUB7 to be more highly reactive to NS1 and cross-reactive. So to summarize the data I've showed you, TUB7 is a flavivirus NS1 cross-reactive monoclonal antibody, which is protective against NS1-mediated vascular leak in vivo and endothelial dysfunction in vitro, and that our crystal structure with TUB7 in complex with NS1 revealed binding to the beta ladder, but then also an indirect steric hindrance to the wing domain, suggesting that TUB7 may be very protective because it can bind to multiple domains. And that finally, TUB7 provides a proof of concept that we can protect against lethal flavivirus challenge against multiple NS1 proteins, which is extremely important for producing therapeutics that can be used to treat any flavivirus infection, a single therapeutic. So with that, I would like to acknowledge everybody in the Harris Lab, including my mentor, Dr. Eva Harris, our collaborator, Dr. Janet Smith and David Akey, who did the crystallography here, Dr. Robert Beatty here at UC Berkeley, who produced this antibody in his class some years ago, everybody in the Harris Lab, collaborators, my source of funding, and you for your attention. I would be happy to take any questions you may have now. Thank you very much.